bets would have changed in the last few months. And so it looks like even though connectomes are a much more high-dimensional object to screen, and it looks like you're going to take a big hit with alpha inflation or error inflation, it looks like they encode important things for the brain, much more so than the volume of a structure or the FA at a certain point. And so even though you take an enormous hit screening a network for genetic association, there's a deliverable payoff, which is much, much higher p-values. And just as we end, I'll give you one example. This is work by my student, Emily Dennis, and Emily works with Susan and also Morella DePreto and others here on autism. And this is a study that shows that carriers of an autism risk gene, it's the contactin gene, these people are healthy. I mean, we know that autism lies on a spectrum between very mildly affected to very severely affected, and it's thought that there's a subclinical expression of autistic traits in all of us at varying degrees, some of them very, very, very minor. And this picture basically says when there's a circle around a node, that is the p-value for the association with this gene, and the quantity being measured is how isolated the network hub is. And so you're all familiar with connectomics. There's all these measures of network connectivity, modularity, small worldness. One of these is called eccentricity, and it's a measure of the isolation of the hubs. So you can begin to do a lot of systems neuroscience by knowing some genes that are good candidates and probing very, very powerful brain phenotypes to see if they have any measurable impact on the brain. So just to end, the number of authors that contributed to this talk is 207. So I won't read out the credit there. Most of all, my student Jason Stein did a lot of work on this, and many of the other folks that you'll see pictured here, except for my daughter, we're doing a lot of the algorithm development for the study. And I do want to thank, I mean, this requires efforts across the world, and the funding agencies in Australia and Europe as well as the US have contributed. Now there's one funny thing about ENIGMA that makes it very different from any other study, and I'll conclude with what it is. If you wanted to do an analysis of a very large amount of data, you'd assume you'd have to collect it in one place. So is there somewhere at UCLA with 20,000 scans? Well, there isn't. It isn't at UCLA at all. What we did for ENIGMA, and it seems to be a reasonable model, a little bit like Wikipedia. So you can work on Wikipedia whenever you like, or if you don't want to, you don't have to. It's just updated all the time. So all of the software for ENIGMA was sent out to the sites that were contributing sites to ENIGMA. And then they did their genome-wide association scan and sent back their results to us. So most of the work in ENIGMA happens, there's no central coordinating center except at the very, very end. All of it's happening all the time at different centers around the world. And so the ENIGMA website keeps being updated with evidence for genetic association with brain measures such as fraction isotropy or connectomes or PIB or other things like that. And it's a very financially efficient thing to do. Now, I mean, maybe you'd take a different view if you were paying for it, but we don't have to win a grant to scan 20,000 people. I mean, no one would pay for it. Instead, you would send software out and you have to have people agree to what they're going to do. But to get one of these projects off the ground, it's more of an act of faith that an individual center alone won't have enough power to discover genetic variants that will be believed. And I mean, that just happens to be the case. When you have around 1,000 scans, you're around the level where you can find genome-wide significant variants. But if someone has a cohort with fewer than that, they pitch in the data to ENIGMA, but no genomes and no scan data ever travels to us. It's all remotely on servers. So with that, thanks very much for your interest and patience. Thanks very much.
Who's got the questions? So I'll start off. So I actually hadn't seen your data on the transferrin HFE marker in blood. Sure. Have you seen any associations with basal ganglia in those? Have you had any chance to look specifically?
Absolutely. So Mark's referring to a very interesting story about iron. So iron is sort of a rogue mineral late in life, and Mark's done some work on manganese and other trace elements. There's a number of metals where actually, is Pamela here? Well, there we go. Pamela's done some work on manganese. It's terrific as well. A lot of these rare minerals accumulate in the basal ganglia and other parts of the brain late in life and cannot be cleared and produce oxidative stress and neurodegeneration. So the notion that some of these minerals, I mean, a lot of people, if you look at a quiz show in the 1950s, there'd be a commercial for Geritol. And Geritol is vitamins with iron added. And it says, avoid tired blood, take Geritol. Well, what you're doing is you're piping your blood with iron. Now, it used to be thought that piping your blood with iron was a very good thing. You'd avoid anemia, and there'd be all sorts of good things. And in fact, for some people, it's good to take iron supplements. But one of the problems is you need iron early in life because it makes myelin. And so the myelin coating on these DTI scans, if we can call it that, was FA, was more intact or higher FA in people with high iron. So that's good. It's a good sign of good transferrin, good iron action in the brain. But the opposite was the case at the other end of the lifespan. And so if you're old, higher iron levels are usually a sign of greater degeneration. I think, Mark, probably the same similar for manganese and some of these more toxic elements. So this is an interesting story. I mean, it does have public health relevance because a lot of how much iron gets into your brain depends on how much you eat through leafy greens or steak or things like this. But some of it depends on your genotype. And if you have a buffer, if you have a gene that sinks the iron out of your blood, and the people with one variant of HFE have a very efficient sink for iron, you'll have very different profiles of brain structure than people with a different iron gene. And it's a very fascinating story. It's a constituent of myelin, that's why it makes a big difference.
Fascinating talk, Paul. I was just wondering your thoughts. Over the past decade, the field has really seen this explosion of studies looking at the relationship between candidate SNPs and brain structure or function in studies that individually have 15 to 30 subjects. And there's been some convergence, and certain candidate SNPs seem to sort of show up again and again as having effects on certain brain areas. But with the advent of these larger scale GWAS approaches, and in the one example you showed, some candidate SNPs like BDNF that we thought would be important seem to not really pan out. Do you imagine it's going to be the case that much of what we've learned about these sort of hot candidate genes that sort of become trendy for a few years, it's going to be wrong? I know you showed the TOM40 seem to hold up, but do you think much of what we've concluded is going to be sort of inaccurate?
I think that's a great point, because if you're statistically very rigorous and you don't find something, you can't claim it isn't there. Although when there's 20,000 people and it's not there, it's a pretty good power of the study. And one way that we think about this, so if a gene is in the news and we want to test it on brain images to see if it has any effect, there's two things that you have to think about as to whether it's going to work. One is the minor allele frequency. And so for all of these genes I've talked about, the bad variant only happens in sometimes 2% of people. So there was one in the news that protects you against Alzheimer's disease. It only happens in 0.5% of people. And some of these, like FTO, the obesity gene, it's found in 46% of people. Now, the reason I'm telling you about that is, if you find a statement about a gene, and the adverse variant is found in 1/2 of people, you've probably got a pretty well-powered study, because half of your cohort is going to be contributing to the effect. If the minor allele frequency, and you can look it up, there's something called SNPedia. And so if you get a paper to review on a gene, you can say, well, that looks like only about 2% of the world has this. So you can see a study with 100 people, and maybe 2 people have the gene. So that's one way of looking at it. The other thing is, you've got to say, well, how big of an effect do we think these genes have on the brain? And so probably the biggest effect we know of is ApoE. Alison and I have studied this a great deal. Probably a lot of people here have studied ApoE. That's kind of the big hitter. I mean, ApoE has a lot of effects. All the other genes that we know, they tend to affect the brain measures by, if you're lucky, 1.5%. So you can do a standard power analysis where you plug in the sample you have, the effect size that you really believe you might get, let's say 1.5% if we're lucky, and then also the minor allele frequency, which is how many of your cohort is going to carry the gene. And you can basically, before even doing the study at all, decide whether you're likely to find a result. And there's a couple of stories that are a little bit in the confusion zone. So BDNF really did pan out a lot for working memory. So maybe we just don't see on imaging the particular thing that the gene affects on working memory. I mean, it could be that imaging is not very good for tapping into certain processes. The other story is COMT, the big psychiatric, and the field has gone back and forth as to whether that gene is highly relevant or not quite so relevant. So I guess to answer your question, if you get a paper to review with a very small number of people, it could be true, but you can say to the people, well, what was your minor allele frequency? What was your power to detect an effect of this size? And some of the effect sizes are not really believable. I mean, I had a paper that I reviewed, that said that one genetic variant, it wasn't a gene that anyone had heard of, caused your caudate volume to be 11% smaller. Well, yeah, it just doesn't sound very reasonable. Now, if that were true, in these genome-wide scans, as soon as you got to that gene, you'd found a big dip in caudate volume. So there is something in genetics called winner's curse. And winner's curse is where you have a fantastic, fabulous finding, and then nobody else finds it with the same effect size. But it's because of the selection bias for studies. And so let's say you randomly did a study at 20 centers, and the people that got the best effect size sent it to science, and the 10 groups that got the worst effect size just gave up. You're going to initially have very high effect sizes. And so I think it's a mixture of winner's curse, and I mean, maybe just knowing what the sample sizes are that are needed for these things. It tends to be pretty big.
Yeah, I mean, if labs are doing voxel-wise searches within their sample, then they're bound to inflate the true correlation between the gene and the effect. So. Other.
Very interesting talk. I just had a question about blocks of SNPs and whether or not there's, at first I would think it would be harder to find a pattern of SNPs as opposed to looking at single candidate SNPs and associations. But when you do see these blocks or haplotypes, is that, do those tend to have a greater effect size on the associations? Okay.
Exactly. So I didn't talk about this today, but that's a very big field in genetics. So doing polygenic testing or multi-SNP testing. I mean, there was an example of it at the beginning of the talk where a whole bunch of genes together were predicting DTI, but I think you're talking about something a little different. If you had a whole sequence of nucleotides that are varying in all kinds of different ways, could that piece of data be predictive? And funnily enough, the first talk today that was talking about ridge regression and shrinkage estimators and that kind of thing, all those methods are used in genetics. And so if you have 100 bits of genome that may have nice signal in, a lot of these penalized regression models, I mean, if you put 100 regressors in a regression, it probably breaks, it'll fail. But these penalized regression models are often used, and you'll see in genetics terms like lasso and elastic net, and I mean, they all come from having a very large number of predictors with small effects. Now, the reason we don't use them in ENIGMA is you could never get anyone to agree which one to do. And so it's like SPM. Everyone will use SPM because everyone understands it. So even though polygenic testing is more powerful, it'd be very difficult to get a consensus about how to do it. But I think, as you said, I mean, that's a much more principled way of looking at genetic variation because you're not looking at sort of one dot in the genome.
Thank you.
I think I'm going to go ahead and move on to people's break. And thank you so much, Paul, for being here. What a treat for all of us.
Okay, thanks so much. Thank you.